Madeleine will be speaking with us about ensuring a future for corals through assisted evolution. So Madeleine was originally trained in marine ecology and developed as an ecological geneticist and began to study corals in 1997 and coral-associated microorganisms in 2000. Her microbial studies were initially limited to dinoflagellate endosymbionts, but in the past 5 years she's ventured into studying other microbial groups that inhabit corals, including prokaryotes and viruses. Her current research focuses on the field of coral reef restoration, in particular the development of coral stocks that may be better able to cope with disturbed environments and predicted future ocean conditions. This includes the manipulation of microbial communities associated with corals, laboratory evolution of algal endosymbionts, selective breeding of corals, and the conditioning or transgenerational acclimation of corals to predicted future ocean conditions. Recently, she began to explore synthetic biology as an approach to increase climate resilience of corals. She completed her PhD on molecular biogeography at the University of, oh geez, I'm going to butcher that, in the Netherlands. And of course I'm in South. Yeah, do you want to say that one for me? Groningen?
Groningen.
Okay. In 1995. Okay, there we go. Subsequently conducted postdoctoral positions in East Anglia in the UK and at James Cook, looking at molecular relationships in the genus Acropora and genetic diversity and specificity of acroporid coral dinoflagellate symbioses. She then took up a position at the Australian Institute of Marine Science in Townsville and commenced as a professor in the School of Biosciences at the University of Melbourne in 2015, while maintaining her part-time position as a senior principal research scientist at Ames. So today she's speaking with us about assisted evolution. So Madeleine, thank you so much for your time, and we'll pass it over to you.
Thank you very much, Rebecca, and thank you for giving me the opportunity to speak here today. And I'm sorry I couldn't be there in person. It would have been very nice because it's very cold here at the moment. Hold on, so I can't, well, that slide seems to be stuck now. Okay. I suppose to this audience I don't have to talk very much about the current rapid decline in coral reef health and coral cover across the world due to both global and local disturbances, including climate warming and acidification, cyclones and storms, terrestrial runoff, crown of thorns starfish, overharvesting, poor development, and so on. So we see across the globe a change from high coral cover and other coral reef organisms diversity to bleached reefs, increase in disease, and basically mortality and a loss of a coral reef structure. And while at a small number of Indo-Pacific locations there has been some evidence that consecutive temperature anomalies have led to adaptation to those conditions by some coral taxa, generally that is not the case, and this has raised a concern that the pace of environmental change is just too rapid for a natural adaptation of corals to occur. And that means that human interventions that increase coral environmental stress tolerance are really very urgent if we want to ensure the persistence of coral reefs as we know them today into the future. And one way that we are exploring in my lab that we think that we may be able to achieve this is through an acceleration of naturally occurring evolutionary processes. And we refer to this as human-assisted evolution or simply assisted evolution. And assisted evolution is really around us in everyday life because most of the food that we eat has been selectively bred or modified in some other way. And even things like our pet dogs, they have been selectively bred for their external appearance and even their personalities. So it's very common in the more sort of commercial sort of areas, but very rare in conservation and restoration initiative, although there are some good examples from the terrestrial environment. So in 2015, we proposed that we should follow a similar approach for corals. And in my lab, we are currently basically doing the proof of concept for the 4 approaches that I've got listed here. So one is, an induction of acclimatization, so basically a preconditioning of corals within and across generations. A modification of the composition of the communities of microbes associated with corals, and that includes the algal and the symbiont Symbiodinium, bacteria, archaea, and we also started to work with the fungi at the moment. We're working on selective breeding approaches, and also we are evolving at the moment only Symbiodinium in the laboratory and then putting it back into the coral, but we're hoping to expand it to the bacteria as well. So today I'd like to give you a few examples of this. It's really in the early stages, this proof of concept work of the last 3 areas listed here as points 2 to 4. And I will start with the selective breeding. So here we use 2 approaches. We breed by conducting interspecific crosses, so we're basically breeding different species of coral. And we're also working with intraspecific hybridisation and assisted gene flow. We're very fortunate to have access to the National Sea Simulator at the Australian Institute of Marine Science, because it's a very large and fancy aquarium facility. Some of you will be familiar with it, where we can simulate natural conditions. So for the interspecific hybridization, I'll talk you through one experiment that we have done, and we haven't completely analyzed all the data, but I have some data that I will share. And in this particular experiment, we crossed 2 species pair, Acropora tenuis and Acropora loripes, as well as Acropora florida and Acropora sarmentosa. And each of those crosses led to 4 experimental groups, two purebreds and 2 hybrids. We did the hybrid cross in both directions. And it's quite interesting to realize that Acropora tenuis is an early spawner. It spawns on dusk, and Acropora loripes spawns much later at around 9 or 10 o'clock at night. But we still were able to keep the gametes and cross these 2 species. Some of the traits that we measured were fertilization rate and the rate of embryonic development. And we then settled these larvae on ceramic plaques. And so it's not actually, of course, the recruits that were colored, but we used colored ceramic plaques so that we could use a different color for each cross, and then randomize the plaques within PVC settlement trays, as you can see there. And we grew them out under ambient and elevated conditions with 12 replicate tanks for each of those conditions, following RCP 8.5, basically unmitigated greenhouse gas emissions. We measured the rate of Symbiodinium uptake, the photosynthetic efficiency of the Symbiodinium, and mortality and growth. Now this slide shows the rate of fertilization of all of these crosses. And you can see that basically both the purebred and hybrid crosses performed very, very well. There was no difference, with one exception, and that was the hybrid cross where we used Acropora loripes eggs and Acropora tenuis sperm, which was a little bit lower. These embryos initially developed more slowly compared to the others as well, but they caught up quite quickly after that. And this slide shows the survival rates of these recruits under both ambient and elevated conditions over a period of 7 months. And it's quite interesting to see that cross that had the lowest fertilization rates, loripes tenuis, actually has the highest survival rates. You can also see that in general, and it's most evident under the elevated conditions, the hybrids perform better compared to the purebreds. Now in this next slide, we've pooled all of these crosses, so irrespective of species, and we just compare the hybrid compared to the purebred corals. And again, we see that both under ambient and elevated conditions, the hybrids outperform the purebreds. Now, what we think what's going on is that, of course, when you hybridize 2 coral species that have relatively divergent genomes, you instantaneously create new gene combinations and new genetic diversity. So we then let the environmental conditions basically select those phenotypes and underlying genotypes that perform best. Everything else will eventually basically die. So I'm thinking it's maybe not necessarily the fact that they are hybrids that they're doing better, but it's just we provided a new substratum for natural selection to operate on, and some of these genotypes do very, very well. The difference in growth were a little bit less clear. I just listed some of the things that were statistically significantly different. So at 7 months, we saw that the purebreds of Laurapis and Florida, as well as one of the hybrids, were negatively affected by elevated conditions. On the ambient conditions, growth of the hybrid Sarmentosa Florida was greater compared to the Sarmentosa purebreds. And then I think also quite interesting, I'll just show you here the size of the recruits at 7 months of one of the crosses, the Florida and Sarmentosa cross. And if you look at the elevated conditions, for instance, you can see that the median size of all these recruits is the same. But the spread of the variance is actually quite different. And amongst the hybrid crosses, we have some of the largest individuals. And we think that is actually quite important. It comes back to that same point that I just mentioned, that some of these hybrid genotypes seem to be doing quite well. And this photograph of the surviving recruits that we now put back in the general system under ambient conditions. And you can see in this particular cross, the Florida purebreds on the right-hand side are no survivors. The Sarmentosa purebreds, there are survivors, but they're significantly smaller compared to the two hybrid crosses. So I think it's quite interesting to suggest that perhaps this interspecific hybridization has some value in terms of creating more tolerant stock. Still very early days in proof of concept. We also are exploring the value of conducting intraspecific crosses as well as assisted gene flow, where you perhaps move adult colonies and allow those to breed with the native populations. And on the Great Barrier Reef, translocations are relevant along the length of the Great Barrier Reef because we see a temperature gradient where it's warmer in the north and cooler in the south. So the thinking is, if we perhaps cross northern genotypes with southern genotypes, that might be a way of increasing thermal tolerance into the southern populations and prepare them for further warming. So that, of course, we have to assume that thermal tolerance has a genetic basis, and also, that is passed on, and that these corals interbreed, and that there's no negative effects on other fitness traits. Now, when we actually moved adult corals from the central Great Barrier Reef to the southern GBR, so from a warmer location to a cooler location, here in this plot at the top, the bar graph shows the temperatures across summer at these two locations, showing that the magnetic island location has more days at the higher temperatures compared to the southern location. We found that during the summer and winter, when temperatures are low and the temperatures go below the long-term temperatures that the magnetic island corals experience normally, they start to bleach, and we saw quite high mortality. And so that means that perhaps assisted gene flow, where you move adult corals around and hope that they will eventually interbreed with the native populations, might not be so effective at this spatial scale on the Great Barrier Reef, because we will lose a lot of that translocated stock before they are even able to interbreed with the native stock. So then perhaps if we bred those corals ex situ and then place the early juveniles, the recruits, back into the field, that might be a better strategy, because often acclimation during development is very important in setting the physiology of the coral, and so we explored this as well. And what we did is we brought in corals just before spawning. We brought in vacant corals from the central and the southern GBR. In this experiment, we worked with corals from Orpheus Island and the Keppel Islands. And first thing we did is we crossed them, and we could show that these crosses had very high fertilization rates. There was no difference between the purebred and the hybrid crosses. And we then we settled these corals in the lab, and then we placed them back into the field, and we followed their survival over time. And in this graph, you can see, so we placed them back in the southern location in the Keppel Islands. And the Keppel Island purebreds had the highest survival, the hybrids had intermediate survival, and the Orpheus Island purebreds had the lowest survival. So that was sort of what we expected. There was no difference in the growth of these different treatments. They were all the same. Now, what we had planned to do is to bring these recruits back into the lab and examine whether they had different thermal tolerance, whether we had bred increased thermal tolerance into the hybrids compared to the purebreds from the Keppel Islands. But we were very unlucky with this experiment. We did this experiment 2 years in a row, and both years we had extremely wet seasons, and we lost all juveniles due to a freshwater plume. So that was very unfortunate, and we could never test the impact on thermal tolerance. Luckily, my colleague Line Bay and her colleagues from Texas University conducted a similar experiment from slightly different locations. They used corals from the far northern Great Barrier Reef and the central Great Barrier Reef, and they conducted the same crosses in the lab and examined thermal tolerance just in the larvae, not in recruits or adults, but just in the larval offspring. And what they found, you can see it here on the right-hand side, is that when corals from the northern GBR were crossed in the laboratory, the original hybrid larval offspring showed higher heat tolerance under the laboratory conditions compared to the purebreds from the cooler locations. And this was particularly the case if the mother was sourced from the warm locations. So this suggests that, this may be a way of increasing thermal tolerance in cooler locations is by breeding these corals ex situ and then placing them back into the field. One concern, though, if we conduct a translocation and perhaps if we breed between these spatially very divergent populations, that they might be maladapted to other conditions, environmental conditions that we haven't measured. And if you're thinking about moving adult corals around, there's an increased risk of moving parasite or pathogens from one location to the other. So we also started to explore basically the scales of local adaptations and what are appropriate scales of assisted gene flow. And we were quite interested in this instance to compare local adaptations between habitats on the same reefs, because adjacent habitats can have very different environmental conditions. For instance, the reef flat compared to the reef slope. So here we sampled 24 coral colonies from each of 4 sites on Heron Island. 2 of the sites were on the reef flat and 2 on the slope. On the right-hand side, you can see we actually measured in situ the temperature and light conditions at these sites. And while the mean temperatures were very similar on the reef flat and slope, the variance was much larger on the flat. Light levels, of course, were much higher and also more variable on the flat compared to the slope. We didn't measure PCO2, but from other published studies, we expect that that varies much more as well on the flat compared to the slope. We conducted a reciprocal transplantation between those sites, and we did a whole raft of measurements, physiological measurements, and also assessed genome-wide SNP variation between the flat and slope populations, and assessed the prokaryotic and Symbiodinium communities using amplicon sequencing of the 16S ribosomal RNA gene and the ITS2 region in Symbiodinium. I'll show you some of these physiological results here. You can see mortality over time since the reciprocal transplantation. And generally what we see is if we transplanted within the same habitat, mortality was quite significantly lower compared to transplantations between habitats. But at the same time, you can see that there was quite a large difference in the response at the 2 locations, the coral canyons and the coral gardens. If we plot these results in a reaction norm, and this becomes quite evident that there's quite a large genotype by environment effect. So the corals actually do best in their native environment, and at the same time there's a location effect. At the end of after about 2 years, we brought the survivors of these transplantation experiments back into the laboratory, and we assessed their relative thermal tolerance. So here I've only plotted the maximum quantum yield of photosystem 2 of the Symbiodinium, which, as most of you will know, is a commonly used measure of health of the corals, and at ambient and 3 elevated temperatures during this laboratory experiment. And again, you can see here it's quite interesting. So for instance, the blue triangles indicate that the source of the genotypes was the slope. So for instance, if we look at the 29-degree treatment, we can see that slope genotypes that had been living on the slope for a long period of time, they had been translocated to their own location, had a certain sort of negative impact of high temperature, whereas the slope genotypes that had been sitting at the flat for about 2 years had to some extent acclimated to these different temperature environments. So they were less affected by this elevated temperature compared to the ones that were not acclimated to these different environments. So there's some recovery of negative impact of temperature by long-term acclimations to the flat environment. Yet at the same time, the slope transplant to the flat environment did not sort of behave exactly like the slope, like the flat genotype. So what these results basically show is that these phenotypic differences are due to both long-term acclimatization to the environment as well as genetic adaptation. So this slide shows the genotypic differences between these populations based on more than 2,000 SNPs in about 900 loci using the genome scans. And you can see this is a structure plot where the different colors indicate basically the different genetic clusters within this data set. And on the right hand side here you see a PCA of the same data where each symbol is an individual. And you can see that the main genetic structure is between the habitats rather than between the locations. In fact, there was very little structure between locations. So this shows that well, we interpret this to mean that these environmental differences between these habitats have led to genomic differences between these adjacent populations. The distance between these sites was less than 100 meters between the flat and the slope, and a depth difference of less than 5 meters. So very, very close, and we expect that gene flow could potentially be quite high. We also looked at the microbial difference. So on the left-hand side, we've plotted the prokaryotic communities. Unfortunately, one of our sites, there were some technical problems, so we only have data for one of the flat sites, only one of the slope sites and 2 of the flat sites. But you can see that the main genetic distinction, or the main distinction between these communities, was between habitats, but there was also a difference between the locations, coral canyons and coral gardens. On the right-hand side, we plotted the most common Symbiodinium genotypes that we found in these samples, and again, there was a main divide between the communities in each of the 2 habitats. So the environmental differences in these 2 habitats, the corals basically have adapted to these environments, not just by changing their own genome, but also by changes in the associated microbial communities. And this is of course relevant to one of the manipulations that we are exploring, where we modify the composition of these microbial communities. And I don't have time today to talk about the Symbiodinium communities, but I think most of you are well aware of how it's now well known that associations with different Symbiodinium types might lead to quite different phenotype of the coral holobiont, and we can manipulate those associations to some extent in some coral species. But we've also started to explore whether we can manipulate the prokaryotic communities with corals. We know that the bacteria and the archaea are critical for the health and functioning of corals, and we suspect that they play a role in tolerance to environmental stressors, although that is not as well understood as it is for the Symbiodinium. But again, this is one of these proof of concept experiments that we've recently done, where we isolated prokaryotic communities from 4 different corals that we knew had different bleaching tolerance in the field. We then reared Acropora tenuis larvae and settled them in the laboratory under in filtered seawater, so under sterile conditions, and then inoculated them with the microbiomes from each of these coral species, and as a control we used an inoculum which was just filtered seawater. After 4 months of growth in these was just ambient conditions, we compared the microbiome, the prokaryotic community associated with each of these recruits in the 5 different treatments. And you can see here where we've plotted this, that there was a significant difference between these communities. So that means that inoculation, a single inoculation with different microbiomes might actually lead to the establishment of a different prokaryotic microbiome in corals. We haven't yet tested whether this had any effects on their stress tolerance, but at least it shows that you can, over short periods of time at least, manipulate prokaryotic communities in these corals at least. And then finally, I'd like to talk just very briefly about the work we have done on the evolution of Symbiodinium in the laboratory. So here, our thinking was that because Symbiodinium species have relatively short asexual generation times compared to the corals themselves, that we could subject these cultures of Symbiodinium in the lab to extreme conditions to increase their thermal stress tolerance, and then if we inoculated corals with these evolved Symbiodinium, then perhaps the corals would be more tolerant as well. So we did this experiment with a very common Symbiodinium C1 type. It's commonly associated with many corals on the GBR and elsewhere in the world. And on this diagram shows a simplification of the experiment that we conducted. Basically, we had the wild type, which was a monoclonal culture that we kept at ambient 27 degrees. And we grew this under conditions that the culture went through many cell generations. So every cell division, random somatic mutations will occur in the genomes. Most of these will have either no effect on the phenotype or might have a negative effect, so they will be lost, but some by chance might have a positive effect. So I indicate that by different colors in these flowers. So we would have seen some random mutations happening there, were not under selections, and we hope, we assume we end up with pretty much the same population because the conditions haven't changed. But we also subjected many replicates of that same culture to elevated temperatures. And so we assume that beneficial mutants will have been selected and will have overtaken that culture. To test this, I should also say, because the wild type is actually unable to grow at 31 degrees. So we assume that mutations have occurred because this selected type is now able to grow at 31 degrees. So we then conducted a reciprocal transplantation experiment to actually test that adaptation had occurred rather than long-term acclimatization or acclimation. And after a 2-week acclimation period, we then, so at the reciprocal temperatures, we measured a range of phenotypes, some of which that I show here. So let's have a look first at the growth. We can see that there was no difference in growth between the wild type and the selected type at ambient temperatures, but at elevated temperatures, the wild type had negative growth. It was basically dying, whereas the selected type it was slower growth compared to the growth at 27 degrees, but it still had significant positive growth. Same with photosynthetic efficiency. The selected type at 31 degrees was as unaffected as the wild type and the selected type at 27 degrees, while the wild type had a negative impact showed a negative impact of elevated temperature in its photosynthetic efficiency. We also measured the amount of reactive oxygen species that would leak out of the cells, because this is important in terms of the symbiosis. As we know that during bleaching stress, the Symbiodinium will produce a lot of ROS and leak that into the host tissues, where it will cause the negative effects. And we showed that under elevated temperatures, the wild type cells produced a large amount of ROS, while the selected type produced similar amounts of ROS compared to the 27-degree conditions. So these results showed that this laboratory selection of about 80 generations had led to a broadening of the temperature range. It was not just a shift, but the cells actually had broadened their temperature range. So very exciting, because it's a relatively fast process of adaptation. So we then inoculated aposymbiotic coral recruits with wild type and selected cells, and did a similar experiments to the in vitro experiment I just showed you. And surprisingly and a little bit disappointingly, while there was still a very small positive effect of the presence of the selected cells, most of that benefit was lost in symbiosis. So we did this experiment with 3 species of Acroporocytheria. And I plot here the growth of these small recruits as well as the Symbiodinium cell density. And you can see that generally at the elevated temperature, the recruits with the wild type cells had negative growth, whereas some of them, the ones with the selected cells, still had positive growth. But so it was a trend. There was no statistically significant difference. It was just a trend. So there was a lot of variation, and the differences were small. Same with the bleaching, the Symbiodinium cell density. There was a slight delay in the rate of bleaching in the recruits associated with the selected cells, but at the end of the experiment, there was no difference. So we're now following this up by looking at gene expression in these different treatments to see if we can understand why there was such a clear signature of adaptation in vitro. Well, when we put these symbionts back into the coral host, we sort of lost most of that benefit. So it's sort of a bit disappointing in terms of assisted evolution, but it's biologically very interesting, and hopefully we will be able to figure out how we can select better, or perhaps in different ways, so that we can transfer this benefit to the holobiont. So in conclusion, this sort of early proof of concept assisted evolution results, while they're not all perfect, I think they are very promising. And we're currently focusing on a number of sort of projects. One is we are rearing the F1 hybrids to reproductive maturity, so that we hopefully be able to do back and intercrosses in a couple of years. We're very interested to follow up on the spatial scale of assisted gene flow, and what is sort of the smaller scale over which this may have benefits. And we're following through with the development of probiotics, so basically inoculating with bacteria and archaea, using whole microbiomes, as well as culturing prokaryotes, and perhaps making cocktails of certain prokaryotes that we may have selected for in the lab, and using those as inocula. I'm quite interested to identify which coral prokaryotes are actually stably associated with the holobiont, and maybe some of the prokaryotes that live inside the host tissues or are associated with the Symbiodinium might be more stably associated, and that's important, of course, again, if we want to manipulate those organisms. We are working on speeding up the rate of mutation for Symbiodinium. So similar to I suppose the hybridisation work we've done for corals, we have been able to fuse protoblasts of Symbiodinium of the same type. And so we will try this for different types of Symbiodinium. It's called basically genome shuffling. It's where we bring 2 different genomes together in the hope that we can create new diversity. We are following up on the identification of key Symbiodinium genes involved in stress tolerance, because those would be candidates for genetic engineering, which is another area that we're starting to work in, because eventually, if we do understand the genetic basis of stress tolerance, genetic engineering will be a much faster way of getting the same results compared to those sort of more traditional assisted evolution methods. And also, and I haven't spoken about that at all, we're still working on the importance of non-genetic inheritance, transgenerational acclimation. And then just this is my last slide before the acknowledgement slide. Sort of this is a decision tree of that we've published recently of how we see that assisted evolution might fit within coral reef restoration as a whole, particularly where we focus on biological intervention. So we really see that sitting down here on the enhancing the resilience of coral stock used in restoration. And I'd just like to acknowledge funding bodies and some of the main people that were involved in the work that I presented. I must say I have a fantastic group with amazing PhD students and collaborators, and I thank them very much for doing all this wonderful work. And thank you for listening. I'm happy to take questions if there's time.
Do we have any questions from anybody in the room? Yeah? Okay, here's Tom Moore.
Hey, Madeleine, I was just wondering if you could speak to your thoughts on the heritability of some of these traits, especially for the, just the straight stress experiments where you move corals around on the reef.
There's very little known about the heritability of some of the traits, but there's a few studies that show that there is quite significant heritability for thermal tolerance, which is of course a very critical trait in this work. So that is very promising. But beyond that, there's hardly any heritability estimates for corals.
Thank you. I have a question. Just it's more of kind of it's come up a couple of times today. So I just wanted to see what your thoughts were from an ethical standpoint of actually implementing in, first of all, what kind of time scale we were, like time to implementation on something like this, and also what your thoughts were in terms of actually scaling this up and outplanting hybrids that were created ex situ onto reefs, and what, just generally, if you would comment on where you stand with how far out we are to understanding the complexities of this enough to being able to implement something like this.
Mm-hmm. It's a really good question, and as I pointed out, this is really all proof of concept work that we're doing, and the upscaling is a real issue. I mean, eventually we need to look at probably very large aquaculture facilities once we know which approaches work best. If we want to do this work ex situ, then yes, that's a huge engineering issue, I suppose, to upscale it. But I think we work because a lot of the restoration is moving towards the use of sexually produced stock, where some of this work is done ex situ or in nurseries. We can probably combine that relatively easily with some of these manipulations. So I think that is promising. In terms of the scale of implementing it into the field, we will have to look at the connectivity amongst reefs. So I don't think we'll ever be able to explant, manipulate the coral stock to all the reefs on on the size of the Great Barrier Reef or wherever you are working. But if we're clever and we're using source reefs that naturally seed off offspring to other reefs, and we will get natural spread of manipulated stock, I think that is the way to go in terms of scale, and that is more feasible. Does that answer your question? Or?
Yeah, that's great. That's a really good point. I just was more voicing that on behalf of some of the community, because I've heard that raised a lot of times. And there's different opinions of, oh, well, we're already kind of messing things up, so we should at least do it in a helpful way. To know we don't know enough, we maybe shouldn't implement. And I just kind of want to know where you stand on some of that. So
there's a lot of debate, and it's the risk of doing something versus the risk of doing nothing. So all of this will need ecological evolutionary risk analysis and cost benefit analysis. That needs to be started very soon as well.
Great. Are there any more questions for Madeleine?
I have a question. This is Mary Hagedorn.
Hi, Mary.
Hey, Madeleine. I have a question related to using source stock generally from the reef, and all of those animals are going through the stressors on the reef. How do you see some of the natural stressors, whether they're local or global, in terms of warming, et cetera, impacting the pace and future of your work?
Well, I think this work is really urgent given the rate of loss of coral around the world. Is that what you mean, or how we use the natural cycle?
No, I meant given that the source stock that you're using to, for some of these proof of concept
I get you. So we're running out of stock to work with. Yes, no, I think that's why we need to work with. And we are right. That's why we are working with you, because yes, I totally agree. Your work and making those cryo repositories, so we have actually genetic diversity to work with in the end, is absolutely critical.
But I guess more to the point is, and this is just my own sort of insecurities about the reef, is I worry that the coral, like our source corals, the ones that you need to do this work and the ones that I need to get biodiversity from, are so stressed that we will be working with inferior corals that will give us answers that are somewhat odd.
So yeah, I think yes, that is for some years definitely a problem. I mean, we haven't had that issue so far with our experiments, but we will have it this year because we had, as you know, 2 consecutive major bleaching events on the Great Barrier Reef. So we are worried about having healthy stock for this year's coral spawning. So yes, it will definitely impact on what we can do. I mean, I hope we'll have some years in the future where the corals will be able to recover a little bit, at least, the surviving corals for us to work with.